Hello, our cherished viewers, watching us from the comfort of your home. You are welcome to your revision show on Joy Learning. Today, I'll be your facilitator, Sir Kwame Amoateng, taking you through one of the basic topics in biology. Yes, I just use the word basic topic. It's a topic we assume or we expect every student in the science department or in biology class should have a grip of it. Terms, how we mark your papers, what we look out for, so that you don't make common mistakes. So I hope you are ready. We can begin. At the end of our lesson today, I expect that you'll be able to understand or get a grip of the paper you are going to write. It's so important I take all my students through this practice. Each time or every time I come here, I'll do my best after today to pick on one thing that we look out for when we are examining your papers. When you understand the paper itself, it releases the stress, the pressure you go through in the exams hall. It helps you go to which part you think you are so comfortable with, or you have your strength, and can let you score a lot of points. Then from there, we'll come to the basic topic, which is cell. We'll get to understand the concept around the cell. Then we would apply it in trying to identify eukaryotic cells, their structure and function. Then also able to distinguish between akaryotic and prokaryotic cells. And then we'll be able to identify eukaryotic cells and outline some of their function if you have time. So I guess you are ready. The whole biology book that you are learning at school or taking through by your teachers is organized around about 8 themes. From here to university, you are still going to use these themes. The first theme is the introduction to biology. It comes with topics such as what is biology, then scientific skills, scientific method, how to write an experiment. You get to orientation, sectioning, microscopes. These are basic things we call the introduction to biology. Then the next basic thing, which is the main dogma of biology, we call the central theme of biology, which is cell. It's for you to understand what a cell is and its relation to why we say something's an organism. From there, you're able to understand the structures, how it works, so that when we get to specific life processes, like respiration, you know what we are talking about. Excretion, you understand it. Movement in lower organisms, you hope you understand it. So cell, at your level, we'll do our best to make it simple. But for those going to write the exams, we'll show you the tricks of what we want to see when you're answering questions on cells. The next one is the life process. All life process. So in our textbook, we have some selected organisms that we treat the life processes. From plants to mammals, to some protists, to some fungi, and to some plants. Almost all the 5 kingdoms are, we make sure that we represent all the 5 kingdoms in any of the life processes, including the controversial virus, who at one time is a living thing, and at that time it's not a living thing. Then you get to diversity, one interesting area where you get to understand you are not the only living thing in this world. You're able to understand how different organisms undergo the same life processes, from tiny things like bacteria, virus, to parasites in your body, to fungi on bread, all those things. You have to understand that we are diverse. Then the next is, once you are diverse, we try to narrow down how most of these organisms interact with nature. This is a topic that deals with a lot of adaptation. You go to the seashore, how do organisms adapt? To the forest, how they adapt? You, as the human being, how you adapt? your brothers or friends like the apes, the chimps, how they also adapt. So this is one broad topic. Then out of it, we try to narrow down human beings so that we look at how we interact with our environment. For example, some cycles, how we interact with microbes, conservation, pollution, treatment of fecal matter, water bodies, and so on. From there, then we get to one topic that students seem to struggle a lot: genetics and evolution. It actually starts with DNA synthesis, then you get to protein synthesis, then you get to cell division. And you build it up to the genetics, the variation, the evolution. Then you understand where human beings are at this time and where we could be going. And the last topic that seems to feature almost over 5 years now is biotechnology. Of all that you studied, we just want to know how you apply the knowledge of biology in different industries. So like pharmaceutical industry, the fish industry, bakery industry, brewery industries, whatever industry. We want to see how you're applying it. In the genetic technology industry, cloning, and all those things, how to make drugs. That's the aim of biotechnology. Don't think we, the examiners, don't like you. After studying of 3 years, we want to know if you actually grabbed what we were teaching you. So our exams is structured in a way that there's a knowledge part. It's one of the easiest things students usually answer. So you categorize the questions in order of strength. And each question, the way they ask you, will require how you also answer it. So at the knowledge stage, we mostly will ask you identify something, define something, describe something, what is, list the following, list 4 things, name something, match this, state the functions of. These are straightforward. It's the one that doesn't confuse a lot. And if you look at our paper, well, we ask a lot of these ones to make sure majority of you can get the marks to cross the failure point. So you can get your C6 or anything. Then you get to the understanding part. A part that we expect that after having knowledge, you can connect some of what you've been told. So we ask you to interpret things, explain things. Maybe explain the role of this, explain how this works, then compare, give example. In biology, when we say compare, we are trying to say take 2 things, look for the differences, at the same time look for the similarities. For example, I give you a crab and an insect. As a good science student, you're supposed to identify some things that are same, or let me use the word similar, and things that are different. Hardly do they ask on annotated diagram. Annotated diagram where you draw something, you label, and you try to write a function of that part you've labeled just in a bracket under it. It's good for personal studies. Then how to summarize things, predict consequences based on a trend. So you just look at the 2021 NOVDEC, you find out that they asked a certain question about antelopes and tigers. There is such a question. This is where now we are turning our attention to. We want you, the student, to think rather than just to
of no or confusing road signs can be likened to driving in a dungeon.
Again, for ladies girls, one of our judges scored 8 for that ball for them.
Schools in Ashanti are ready to battle for supremacy and the bragging rights as the best school in debate. Can St. Monica's Girls Senior High School defend the title, or will any of the 15 schools including OWAS, Prempeh College, Yasantua Girls Senior High School, TIAMAS, Presby SHTS, Bompata, KNUST SHS, Tepa SHS and the like wrestle it from them? Make a date from Friday 17th June to Friday 15th July 2022 as you get thrilled to fierce and insightful arguments in the Love FM High School debate from senior high schools in Ashanti. Catch the action live on Facebook at LUV 99.5 FM, on radio on Love 99.5 FM, and on Joy Prime and Joy Learning on Monday to Friday at 2 p.m. each day at the Christian Service University College. This event is brought to you by your superstation Love 99.5 FM in collaboration with the Ashanti Regional SRC, the Ghana Education Service, and the Conference of Heads of Assisted Secondary Schools, CHAS.
The rainy season is here again. In the major cities of Ghana, flooding has become the key source of human vulnerability, and varied attributions have been given to its frequent occurrences. Dear learners, it's time to show off your writing skills on the Joy Learning Essay Club contest. In not less than 300 words, write on the topic, what can be done to prevent perennial flooding in Ghana's major cities? Your essays must have your full name, your age, your contact, and your school's name. Send a copy of your essays to Facebook at Joy Learning TV. You can also email to joylearningtv@myjoyonline.com or drop off at the front desk of Multi TV office. The best 10 essays qualify to be part of the finalists of this educative contest, and there are so much Joy Learning branded items to be won. Not forgetting the opportunity to become a Joy Learning ambassador for 2022. Remember, the topic is what can be done to prevent perennial flooding in Ghana's major cities, and the deadline for submission is Friday, 24th June 2022. Joy Learning Essay Club, improving our writing skills and sharing our thoughts on national issues. Joy Learning, keep learning.
All of you, I could see you were so engrossed with what was going on. We apologize for the break in transmission. Let's continue and pick up from where we stopped. Now, I got to the point of application in the exams. They want you to try to demonstrate that whatever we teach you in class, you've picked it, you can use it to solve a problem. Sometimes it can come in an experimental form, that skills that we taught you, how can you use this to filter something? How can you take care of maybe refuse in your environment? So we want you to demonstrate to us that you can solve things based on models and principles and theories of biology. Now, you go to analysis. Know that from knowledge to understanding to application to the level keeps increasing. So there's a way we award marks to each question. So just sit down, look at questions like solve or state the theories, explain this, describe this. Just look at the marks associated with them. It's sometimes higher than when we say list, state, name. In the analysis, we want you to be critical of things by knowing your biology well and using it to analyze or to differentiate things, compare things, to infer and draw some point, and also make probably a prediction on something. The innovation and creativity is what we term also as synthesis. We've given you something, and let's say we ask you, if we give you this medicine and you take it, how will it work on, let's say, Plasmodium? So there we want you to demonstrate to us that you know the drug, probably you know the life cycle of Plasmodium, and which stages will these work? So innovation and creativity also looks at how you can think outside the box. Like recently, just ended, we asked something on poaching on certain parts of certain animals. And we try to test your mind about what goes on. So the last is the evaluation, where we try to, it's a little bit, we say one of the most difficult stage, where we try to let you come up with maybe an experiment, hypothesis, justify something, compare features of different things, or give a reason to support something. These are the advanced form of our exams. Now, with this knowledge, let's go to the papers you are going to write. In the exams hall, you have two papers that will come together. That's what we call the theory paper. That's how we call it. We have paper 1 and paper 2. And we used to call the biology 1 as the objectives. That's how we call it. It's a questions of 50 questions in all. 1 to 50. Whilst the paper 2, you have the first 4 sections called the section A. First 4 questions of section A. It's for all students in the West Africa zone to choose 2 questions from section A. But the section B, we have it in 2 parts. One for Ghanaian candidates and one for other countries. As a Ghanaian, make sure you only answer question 5, the Ghanaian one. Don't attempt to answer question 6, which is for the Nigerians and others. We won't mark it. You won't get any mark for that. I repeat, you won't get any marks for that. So, on your screen, I've highlighted the section A part. It can help you usually know where to go when you're in difficulty in the exams. The first 9 questions is what falls under what we call the basic biology, as I explained earlier. Chapter 1, cell, osmosis, classification, all these ones fall under the basic biology. The first 9 questions. Note this in exams too. Usually when you start from each block, the first 3 questions are sometimes very easy. As you go up, it becomes a little bit difficult or tricky. And usually, they kind of give you long-worded questions. What we are advising is that in your first 30 minutes, just look for the easy ones and pick your marks. The next 10 to 27 or 29 questions, that's question 10 to question roughly 27 or 29, depending on the year paper, is a mixture of plant and mammals. It's always a mixture of plant and mammals. If you think you are not good at it, jump. Or look for the easy ones within it. Usually those that say, identify which of the following is this. Look for those ones, answer it quickly, leave the long-worded ones down, and go to the next stage. The next stage on question 27, 29 to 38, this is where we have the interaction in nature, man and environment, which we just described earlier. It also starts from some of the easy ones. First 2 or 3 are very easy. As you go up, they start giving you the applied one. Question 40 to 47 or 48, usually DNA, genetics, evolution, variation. That's how they ask the questions. So from there to look for easy ones. Which of the following is a pyramidin? Which of the following is maybe a base of that? What is the chromosome number? These are the easy ones. Pick them quickly, then go back to the difficult one. Question 48 or 49 is so funny that every year comes from an insert question. You can take all the past questions and look at it well. Around 48, 49, usually it's an insert question, mostly on social inserts. Usually comes at 1 question. So as I'm telling you, just if you know your biology well, go to the easy place, spot it quickly, get your marks, and just move on. Question 50 usually is on evolution. Either 49 or 50 always is on evolution. either the theories or the evidence. This is how our paper is structured, and that is paper 1. Now, what we call the section B that usually we write, it's divided in such a way that the first question 1 and 2 is a mixture of almost entire biology. Less ecology, man and environment, genetics, DNA. So what they usually do is that we look for a concept in biology. Let's say excretion. We start asking what's excretion. Then we zoom in either into plant or animals. Then we start asking a little bit in-depth question. And usually like comparing something in plant and in mammal, or taking two organisms and comparing them. That's what mostly we do. Or we'll give you some of the basic like diversity question on classification. This is how we structure it. But for question 3, most students try to narrow down question 3 and 4 because they choose 2 questions out of 4. Those who are so good in the ecology, or what we call interaction in nature, and man and its environment, usually will go to question 3. As much as that question usually is easy, the use of our technical words, which lose or confuses the normal way we speak English, usually gets students into trouble. And I said each day or each time you come around, I will try and always narrow down one area and give you the tidbits over there. On question 4, some also narrow down there, which is a DNA question, a cell division question, genetics, evolution, variation. If you are good at it, you go to question 4. As I said, we will show you the tricks later when it comes to the crossing. Why a student will get 9 marks, somebody can get 0, although they are all the same. I can get 0 easily, or you can get 9 or 18 marks there. Question 5 is a mixture of all the themes. It's compulsory for Ghanaians to answer. It carries 30 marks. The other ones I spoke about carries 20 marks each. If you choose 2, that will give you 40, plus the 30, giving you 70 marks. And the section A is 50, giving you 120 marks. I've said don't go to question 6. It's for Nigerian, or the Nigeria, for candidates from Nigeria. Don't go to that area. So they also don't answer Ghana questions. Now, question 5, what is usually unique about that is the application questions, usually from biotechnology. Each year, they ask a question either on fuel, preservation, water treatment, or fish stock, anything that they want to ask. So take note of that area. Then we come to the practicals. Now note this. For students going to write the section B, as usually called the theory questions, it's a rude, they will ask a question and give you lines there for you to write accordingly. It's not the olden way they will give you an extra paper. All the questions they ask you, they will rule lines, and you are supposed to use that space to answer. So you just don't stand up and write anything just like that and start cancelling. We don't give an extra sheet. If you are lucky, there are some few pages at the back. But we advise you to always make use of the lines there. They've designed in such a way that even handwriting are very big, it can still fit in the space there. It also helps you know how many things you are supposed to write and the marks you expect to get. This is paper 3, the practicals. We don't write all these on the same day. We write this just in 2 hours, 40 minutes, one day. Another different day, you'll be called to come to your labs to answer questions using real life, preserved or real life organisms in the lab. This is what we call practicals. That one to question 1 and 2 is for all students in West Africa. Question 3 is for Ghanaians only, and the question 4 is for the Nigerians. Please, for that one too, we have rule lines here. We have rule lines here. First two questions usually carries about 50 marks, 25, or however they combine, it gives you 50, and the last question of your country gives you 30, making sure that you get 80 marks from this. So the total marks you are looking for in all your 3 papers is 200 marks. Make sure you can cross 140. If you can do your best 160 and above, you are safe. I always tell students, do your best, especially section A. Make sure you focus on and get over 40 or 45 over there. Okay. Let's go to today's topic. What I'm going to do is that I'm going to announce the phone lines. Then I'm going to pop up a slide with 6 questions. You are at the house or at home, call in and try to answer as many as you can in the shortest possible time, probably less than a minute. If you are not able, and we pass it on, the next person who calls in, I'll tell you the ones that are wrong, and I expect you to finish that before I give you your slide. So the lines that you can call through is 0302 221 750 or 0302 221 706. So I'm going to pop the first slide of 6 questions. Let's see who can answer this as a way of revision. You can call us on 0302 221 705 or 030 221 706. Appleton. Richard.
Sir.
Yeah. You're welcome.
Thank you, sir.
Yeah. On your screen, where are you calling us from?
I'm calling from Accra.
Where specifically in which school?
Accra.
Yeah, which school?
Sir, pardon?
Which school are you calling from? Which school do you attend? TI, my dear?
Yeah.
Okay. On your screen are some basic biology question on cell. Try and answer as many as you can for us.
Okay. Hello.
Hello, Richard.
Sir.
I'm looking at, yes. Question 1. We lost Richard. Can still call in 03022 21705 or 0. Prince. Yes.
Hey, hold on. Sir.
Yes, sir. Kindly help us with the questions on your screen. TV so that you can listen to us.
Oh, yeah, okay.
Okay. Yusuf.
I'm calling from Kumasi University Senior High.
Okay, Yusuf, thank you very much. KNUST Senior High.
Yeah.
Please, on your screen, are you in Form 3?
Form 3.
Yes, let's do this as a revision. Let's crack the brain a little bit. First 6 questions. The first scientist to describe the cell was? Can you hear me?
John Lewis. Sir, the first is John Ray.
John Ray
is the first scientist to describe the cell.
The second one. Let me make asterisk here. Question 2. The basic structural unit of organization of life is referred to as? The basic structural unit of organization of life is referred to as
It's referred to as cell.
a cell. Okay. The living material of the cell is called dash, and it consists of. So the living material of the cell is called dash. What name do we give to the living part or the living materials of the cell?
It's nucleus.
Nucleus. Okay, let me give an asterisk there. And it consists of what? Okay, let's move on to the next one. The structure of the cell that controls the movement of substance in and out of the cell is the Come again.
Cytoplasm.
Cytoplasm.
Yes.
Okay, let me make an asterisk here. Why is the cytoplasm of the cell considered a very important component?
Because it stores food for the cell
Okay.
and contains
Then try. Then the last question for you is which structures control the activity of the living cell? Which structures or structure controls the activity of the living cell?
the nucleus.
The nucleus. Clap for yourself. Clap for yourself. Though you made some mistakes, let's wait for your other colleagues to also show us what they think. Thank you very much.
Thank you, sir.
Okay. You can still call us on 03022217005 or 03022217006. There were some few things you couldn't get right. So if you come, try and see if you can also get the correct one, then move on to the next slide. Asari Boatin Samuel.
Yes, please.
Yeah, welcome.
Thank you, sir.
Where are you calling us from? Which school?
Please, I'm calling from Kedi of Pope John Senior.
Wow, okay. Okay, let's go to our slide quickly. Question 1. Your colleague couldn't get it. He said John Ray. What do you think is the answer?
Robert Hooke.
Robert Hooke is correct. Then let's look at question 3.
Question 3.
Yes.
Okay, the living material of the cell is called the cytoplasm.
Okay. And it consists of?
It consists of the protoplasm and the nucleus.
Okay. Let me give a asterisk there again. The structure of the cell that controls movement of substance in and out of the cell is the?
It's the vacuum.
Okay. Then why is the cytoplasm of the cell considered a very important component?
Because it plays many roles. I think it contains some structures which helps in the managing of the or protection of the cell.
Okay. Then I think the last question was answered.
Okay.
You've done well. Clap for yourself. Okay. Before we go on to the next slide, let's try and correct some things. Question 1 was right, Robert Hooke. Question 2, as our colleague said, is a cell. Question 3, depending on how it was worded. The right answer was protoplasm, and it consists of nucleus and the cytoplasm. The structure of the cell that controls movement of substance in and out of the cell is our cell membrane. The structure of the cell that controls movement of substance in and out is our cell membrane. And why is the cytoplasm of the cell considered very important? First, it houses almost most of the organelles, and all biochemical process metabolism takes place here. Let's go to the next slide. We're waiting for any of our students to call in and also attempt slide 2 of our 6 questions and see how well you do on your biology. Waiting for you to call so that we do this quickly and see or test your strength on your speed test on basic biology. You can call us on 030-222-1705 or 0302-221-706. Questions that you come across are: the structure responsible for reproduction in a cell is. The organelle involved in the transport of substance within a cell is called what? The scientist who discovered the cell while examining a thin slice of cork under the microscope was who? And cell membranes are composed of. The one important function of the cell membrane is to do what? And the hereditary or genetic material in a living organism is located in what organelle? Okay. Take your time, look at it. Or I'll just go to the next slide. So you choose which slide. This is slide 2. Where do protein synthesis in living cells occur? The network of double membranes that help to convey materials through the cytoplasm is what organelle? The mitochondria has unfolded portion. What is it called? Then the organelle, which of the organelles in our cells store some digestive enzymes for that taking digestive functions? The scientist who discovered the honeycomb structure of the cell was who? And the cell membrane of a cell is said to be semi-permeable because of what? Why do we say a cell membrane is semi-permeable? So waiting for you to call in. Note, this is a revision show. I'm supposed to prep you up, poke you, so that you try to recollect some of your basic biology topics. Okay, seems your calls are not coming through. Let's move on to some questions that involve pictures, drawing for you to choose one of the correct answers. On your screen are basic cells, some in living things and some in plants. They are asking you which of the Kwache. Hello, Kwache.
Yes, sir.
Yeah. Welcome.
Thank you.
I believe you were with us at the beginning and you saw some of the slides. Which slide do you want to go to so that you try your hands on some of the questions?
Please, the 41 one, the one we continued.
Okay. Okay, this one.
Yes, please.
Okay. Let's try your hands and see.
Okay.
The structure responsible for reproduction in the cell is?
The nucleolus.
What did you say? Nucleolus?
Yes, please.
Okay, let me make an asterisk. The organelle involved in the transport of substances within a cell is what organelle?
The cell wall.
The cell wall, okay. The scientist who discovered the cell while examining a thin slice of cork under the microscope was?
Janssen Anton Leeuwenhoek.
Okay, Anton Leeuwenhoek, don't worry. Then cell membranes are composed of what?
The cell membrane. The cell wall.
Okay, you've tried. One important function of the cell is to
protect the cell or the organelles inside the cell.
Yeah, that's correct. But see, they narrow down one important, usually the first important one.
Transport it or it serves as a membrane when or food materials enters through the cell.
You have the idea. And then question 12. The hereditary genetic material in a living organism is located in which of the organelles?
In the nucleus.
Ah, well done, well done. You did your best, you got some correct and some you tried your best. Okay. Let's wait for your other colleagues to also call in.
Okay.
Hello?
Hello.
Yeah. Please, who is calling?
This is Obed.
Obed, calling from which school?
I'm calling from Volta Region, Summer Senior High School.
Okay. Your colleague got some of his answers wrong. The first one was correct. The structure responsible for reproduction is the nucleus. Now, the organelle involved in transport of substance within a cell is what?
The smooth endoplasmic reticulum and then the rough endoplasmic reticulum.
Correct. That's ER. Now, question 9. The scientist who discovered the cell whilst examining a thin slice of cork under the microscope was?
That's Robert Hooke.
That's correct again. Then question 10. Cell membranes are composed of?
I'll say cellulose.
You have an idea? Okay, we'll go back and revisit it.
Okay.
The one important function of the cell membrane is to do what? I use the word very important function.
That's to control the selective passage of.
I know you've gotten it. Yes, selective permeable membrane.
Yeah.
It regulates substance that has to enter and leave the cell. Then your last question, question 12. The hereditary or genetic material in a living organism is located or found in which organelle?
It is found in the nucleus.
Clap for yourself. Clap for yourself. Well done.
Thank you. Thank you, sir.
Okay. The question 10, I think students are struggling with it. Cell membrane are composed of phospholipids. And within it, we have the protein immersed in it or embedded in it. We would go through all these ones, don't worry.
Okay.
Yeah, let one of your colleagues also call through. Thank you very much.
Thank you, sir.
Okay, you can call us on 0302 221 705 or 0302 221 706. On your screen is a slide of 6 questions. Let's see how you can tease your brain and see who can get most of these right. Okay. Okay. Now let's look at how the section B type of questions are framed. And from this point, we'll use these questions that have come so far to teach. Now, one question that they asked, mention scientists associated with cell studies. Mention 4 ways by which component of the cells can be studied. So how do we study the nucleus? How do we study the mitochondria? Then explain why the cell is the functional unit of living organism. We know the definition, but they ask why is it the functional unit? Then mention 3 organelles of the cell that are double membrane, single membrane bound, or non-membrane bound at all. Then the nucleus controls the activity of the cell. Why? Then still 3 structures found in the nucleus membrane. Then also asks you to try to write the correct sequence on the cell organization in an organism. Okay. Let's also look at some section B. So far, we just grouped all the section B questions under cell and how they usually ask these ones. First, they said, try and explain the cell theory. Great. Scientists. Hello.
Hello, sir.
Yes, my boy. How are you?
Sir, I'm fine.
Okay. Let's try to answer one of our questions from question 19 to 24. Where are you calling from? Which school?
Sir, from St. Augustine.
Okay. Form?
St. Augustine. Form
Yes.
2.
Form 2. Okay. I believe that you've treated this stuff. Let's see if you can try question 19. The thread-like material present in the nucleus are known as?
Thread-like material.
Yes.
The mitochondria.
Mitochondrion.
The
Okay, let me put an asterisk here. Then question 20. DNA formation is associated with which organelle?
DNA formation. Nucleus.
Nucleus. The rough ER is different from the smooth ER because of the attachment of?
Say the question again.
The rough ER, that's rough endoplasmic reticulum, is differentiated from the smooth ER because of the attachment of?
Ribosome.
Okay, that's correct.
Ribosome.
The fluid within the mitochondrion is called? The fluid. There's a certain fluid in the mitochondrion. Okay. Now, the presence, now listen to the next one, it's an applied question. The presence of a large number of, it's supposed to be large number of mitochondria in a cell indicates that the cell is what?
Red blood cell.
Okay, I'll explain later. And lysosome contains what? Question 24, yes, lysosome, it contains what?
It contains 3 pairs of.
We can hardly hear you. It contains
3 pairs of triplet tubules.
Come again. It contains tubules.
3 pairs of triplet tubules.
Oh, okay. You're talking about the microtubules in the centriole. Okay. Yeah, you've done well. You've really done well. You've given us your best informed too. Yes. Thank you very much. Let's wait for other colleagues to also share their idea on these questions. Okay, so we're at this stage trying to look at all the questions that had come so far in WASSCE from 1999 or from the Nigerian Quest 1988 up to date. Now, what is a cell? Looks funny. What's an organelle or a tissue? Then they ask, name 4 organelles in a typical cell. In a typical cell. State one function for each of the organelles that you have named. Then describe the structure of the following organelles and state always one function. So first, you are supposed to describe it, tell us all that the makeup is, then what they do. Then in WASI, let me go back. Okay. There's something here I've said in a tabular form. This word, tabular form. In exams, if we instruct you to use a table and you don't use it, it's -1. Keep in your head, once we give you instruction and you don't follow. So when they tell you to draw something 6 to 8 centimeters and you don't draw it accordingly, you lose a mark. So in a tabular form, state 3 structural difference between a cell wall and a cell membrane. State one function of a cell wall and a cell membrane. What's the main component of an eukaryotic cell? What's an organelle? It has repeated itself. Then difference is the same thing. Then question 5. A diagram. They insist you draw a diagram. It happens, it carries about usually 8 to 10 marks. They tell you to draw it by giving you a specific instruction, 6 to 10, and which side you should draw. Then they say, give the function of 2 labeled organelle. Then they also go on, if they use the word compare in exams, means that look for differences, then come and write something on similarities. Don't only do difference and leave the similarities. You lose marks. Now, state 3 structures that are found in plant cell only and some structures that are found in animal cell only. Then describe the structure in plants which enable them to carry out photosynthesis. Then the last question so far was state 4 structural difference between prokaryotic cell and eukaryotic cell. On your screen is a section A drawing of a plant cell. If you can do your best to join the lines, they are trying to say, try and identify the parts and label them. As much as I won't try to identify everything here, when we do the main study, you get the correct names. On your screen again, problem of the day. This is a NOVDEC practical question, or we call the private candidate papers. This is what they brought in the exams. I, Vin, and stuff. Then describe the structures and state one function of everything. So all the organelles, you should try as much as possible to know the functions of these organelles. Then because it's a plant cell and it's a palisade cell, they asked how are they adapted. And in WASI, when they ask adaptation questions, what you are supposed to do is that state a structure of whatever they've asked you. An adjective of it, maybe it's elongated, it's rectangular, is that. Yes. Hello. Hello.
Hello, sir.
Yes. Welcome. What's your name?
My name is Philemon.
Philemon. Calling from?
Yes, sir.
Calling from?
Calling from St. Augustine.
St. Augustine, okay. On your screen is a plant cell. If you can identify as many as you can the names of the parts of the plant cell, you will be very happy.
Yes, sir.
Okay.
Okay, sir. I'm checking this from the screen.
Okay. It's not only I, any of them that you can easily label.
Should I answer the question of the day?
Yes, problem of the day.
Okay, sir. The I is self-government. From question 1 or 2?
As I said, the labeled part, any of them that you can identify.
Okay, the V is the cytoplasm.
The V is the cytoplasm, correct?
The VI is the vacuole or the sap.
The VI is the, yes, sap.
And the VIII
Okay.
is the microplasm.
Nucleoplasm. That's, you're talking about V with the 3 eyes there. That's 8.
Yes, sir.
Okay.
And the IV.
Okay. Our colleague has tried his best to identify what he can. Okay. Hello. Hello. We lost our caller. So we'll go on. When we get somebody call through, we'll come back to the problem of the day. There are 2 problems of the day. There's another one of an in-depth animal cell. The first one is a plant cell. I give you an in-depth animal. Look at it, and when you call, you try your hands. Elizabeth.
Yes.
Welcome. Calling us from?
Tamale.
Tamale. Oh, congrats. And for form, you're in what form?
Form 1.
Wow, that really.
SS1.
Okay, you've been taught the cell, isn't it?
No, there's not teachers.
Wow, okay. Can you try your hands on any of the parts on your screen?
Yes. Hello?
Hello? Of the parts I on the drawing.
Hello?
Hello? Can you hear us? If you are behind,
Yes.
it's your volume. Or reduce the volume of the TV. Okay, we lost. So let me take you through quickly in 30 minutes about everything. Now I asked question about the history of the cell. Now the first person we credit identifying the cell is Robert Hooke, who around 1663 first was made the creator of the science, the creator of a science, let me call it something like you can call it a museum, a meeting place for scientists in England. And every week they are supposed to meet and try to do something extra, wild each other with new discoveries. With lenses, and lenses was in vogue at that time, and everybody was talking about lenses. So he tried to also think about what he can do. So he granted his own lenses, made his own microscope, and tried to look at the cork. Cork is the back of a plant. Let's call it the back of a tree. It was fascinating at that time. Solid material. But when they throw it into water, it doesn't sink. So they understand why solid materials that they've proven are all solid may sink. It wasn't sinking. So Robert Hooke has to design his own microscope. Tried to look at it for the first time. He couldn't see it because it was very thick. He has to use his pocket knife to keep cutting layers upon layers off. So you could able to get a very thin slice. That's why we emphasize on thin slice. And in the microscopy, we usually do things in thin slice. Then if it's primitive, first, somebody will say did the first, let's say, light microscope with using lamp as a source of light, was able to see a box kind of thing, and he said he called that box thing a cell. He said it looks like the monk's house, a room with a boundary. Now that boundary in future will come to study and know that there was a cell membrane and there was a cell wall. It was hollow, so the thing could float because it was filled with air. Unknowing to Hooke, he didn't know that this was a living cell of a plant. He first called it cell. Then 10 years down the line, 1675, around the same time, Anton van Leeuwenhoek, he was a wine taster, has nothing to do with science, and he was a clove trapper. Those who look at cloves, they work on cloves. So they have to look for magnifying glasses to look at the lines of the material, to grade it. So later, after using the hand lens, he became enthused about it, started making it a hobby, and they knew how to grind it. All these stories are on our Joy Learning TV. Go to Cell. You have the full details on Cell. I'm just trying to brush through it. Then he made his own, tried to look at things, including things on his teeth, tried to even collect water and rainwater. First he collected it, looked through, he saw nothing. Later, after a week or some 3 days or to a week, he looked through it again and he saw tiny things moving, and he called it the animalculus, meaning tiny creatures moving. Then later, with that, about 100 years later, scientists were fascinated and were trying to work on it. Then one scientist called Robert Brown, a Scottish scientist, observed many different cells and saw there was a dark portion because they were improving the microscope. So you could see that every cell, whether plant or animal, has some dark portion. He called it nucleus. Then another scientist called Dujardin, a French scientist, saw that after somebody saw that, he started testing what is in a cell, and he saw that all cells across all organisms has some content or certain liquid or fluid inside with other things. And he called it the sarcode or sarcode, which later we came to call it the cytoplasm. Then Matthias Jacob Sheldon, or that's a German way, some say Sheldon or Sheldon, whatever you call it. He was able to propose that after studying a lot of plant cells, that plant cells are made of cells, like all plants are made of cells. Not knowing at the same time, another scientist called Theodore Schwann in 1839 was also studying animals, and he also said that all animals too are made up of that cell Robert Hooke spoke about. Another person also said plants also had the same cell. So two scientists working on two different organisms was able to conclude that all organisms of that lane or that tangent all have cells. Okay, so animals have cells, then plants have cells. At that time, these were the common organisms they were working with. So they just concluded that all living organisms are made up of cells. Then later, Rudolf Virchow, he is a German physician or a biologist who published a lot of cells, a lot of things on cell, concluded that at that time too, in the 18, after the Robert Hooke era, we went into microorganisms, where people were studying microorganisms. They also saw that microorganisms were giving rise to microorganisms. So they scientists, after studying a lot of information from cells, concluded that one cell comes from another cell. And you call it hominia cellula e cellula, which means cell give rise to cell. So remember, somebody have said that there's something called a cell. Somebody has said all organisms are made of a cell. Someone to have said that cells come from cells. These facts are what we put together to call it a cell theory. So the cell theory below on your screen says that all living things are composed of cells and are product of cells Then a cell is the basic thing in all living organisms. For you to be called a living organism, that is the basic thing. If you don't have it, we can't class it. A stone don't have a cell, so a stone cannot be called a living thing. Then, like what Rudolph said, cells arise from pre-existing cells. So when they defined the second point, cell is the basic structural unit, they wanted to explain, or the syllabus wants you to understand, why is a cell the basic thing? Basic because it is common and the most fundamental thing seen in all living things. Every living thing, from bacteria to mold to anything that we call living thing, has that. Then we use the word a unit. A unit means it can exist on its own. It's a discrete item that can house its own thing, have its own biochemical process, survive on its own, and is self-sufficient. For you human being, you may think all cells are in you, but for unicellular organism, everything is going on in that thing. So we say a unit. Then the last thing is that all living things have it. That means a living thing had a cell, while non-living things do not have anything as such. For the Form 3s, I believe that you've been taught parts of the cell, the organelles of the cell. How did scientists come about studying this? We are required to know that we use microscopes, especially the electron microscope, which will give you very powerful images of, and also magnify small things. And that is what we call cell fractionation. In cell fractionation, this is what we mean. An ability to find a way to rupture the cell, grind it using a blender, using homogenization, anything that we can use to grind it so that we can get the content out. We use centrifuge or centrifugation to a certain machine. If you've been to the hospital where they've taken your blood, you see them put it in some round thing that goes round. It has to whirl around so that the cytoplasm, which is a little bit jelly-like, is so sticky that the content can easily settle. So once you whirl it around with force, it will force the content to settle or to drop to the bottom. And it drops according to size. So size and weight. So the first one, probably the nucleus, is the biggest, the heaviest. And the least ones are the ribosomes, which drop last. Then we have something we call microdissection. It's a technique that they put a cell under a microscope. Try to use tools by looking through a microscope. Try to, let's say a pen, try to pick maybe the nucleus out of the cell and leave the cell down and study only the nucleus. If they are done, they can decide to put it back. If they want to study the mitochondria, they take only the mitochondria out, they can decide to put it back. Then we have chromatography. It's mostly chemistry. It is how certain chemicals rise up on a paper. So they use this also to know the content of most of the organelles that we know. And the sixth one is the staining. Staining helps us see certain things we could not see easily to become very clear for you to see. Let's say if you use iodine to stain onion cells, you see that the borders become visible for you to see, and you see the nucleus become a big brown dot under the microscope. So let's go on now with the main lesson. Take your time, look at what's on your screen. On your left is your plant cell, on your right is your animal cell. We've done our best to give you the best description of these cells. Let's start with the animal cells. Look at the nucleus position, which is down here. And the nucleus has things like nucleolus, chromatin, nuclear pore, nuclear envelope. Just look at it. Later, we'll keep going and coming back to look at this. Okay, let's move on. There is a simplified drawing to, or a sketch type. We use the word typical cell. That means you may not get everything in one plant cell, but we've done our best that we brought the little organelles in most plant cells, try to draw it and put in one plant cell. Same in animals. Look at where the nucleus are found in an animal cell and a plant cell. Look at the things associated with the nucleus, just around the nucleus. Look at the shape of a mitochondria. Look at the shape of a chloroplast. Look at the shape of a vacuole. How smooth endoplasmic reticulum is drawn against the rough ER, and how Golgi bodies are drawn. So look at some small vesicles that seems to be associated with the Golgi body, so that when we go back to our problem of the day, you'll be able to identify it. Okay, let's go on with the main study. The first outer structure you see on a cell is what we call the cell membrane. As a revision, which means I won't take time to draw how lipids looks like. That outer membrane that houses everything of the cell is a lipid, on which the top of the lipid have what we call phosphate associated with it. So the two combined molecules, use the word bilayer. You can see that there are two layers, one here, another here. So these two layers is facing each other is what we call the bilayer, biphospholayer. Then in between the biphospholayer, we have immersed or interspersed with it some round structures you see on your screen on your left, which we call the globular protein molecules. Usually, the lipid allow substance that can pass through lipids to pass there. That's the non-polar substances. And the polar substances which can pass through the non-polar will only pass through the globular protein. The way they behave depends on whether the cell would expand energy at that globular protein area, would then let if the cell is using energy or the cell is not using energy. So you have passive and active movement of substance through it. The main thing is that this pore can decide to close, not allowing something to pass through. Or if the thing is too big, it can't pass through. So we say that it regulates the passage of substance in and out of a cell. That's what we call the semi-permeable membrane, semi-permeable function of the cell. Some will say membrane. Now these protein too are classified in many ways. We have the transmembrane protein, integral protein, peripheral protein. You get a full lesson on cell. Now the idea of this lipid that we use was designed or was proposed by S.J. Singer and J.L. We mostly call it fluid mosaic model. It's a simple thing, the fluid mosaic model. It's just like you have tiles in the house and all the tiles have some design. So when you move on your left, when you move one step to your left, you move to your right, you see that the same design is there. If you go round, it's like you keep moving, but you keep seeing the same design. So what happens in the fluid mosaic is that the lipid is fluid. The lipid is like, let's say, oil, as we mostly call it, simple thin oil. It flows, but as it keeps flowing around, the way the protein are arranged, though it will turn around, the same shape that you see at the top will maintain itself even as the fluid moves around, keep moving around, but still the arrangement is kept. It's so unique. If you have the opportunity to watch a video, you see that it keeps turning, but the arrangement keep the pattern, like the way your tiles in the house keep its pattern. And that's what we call the fluid mosaic model. Now, in some organisms, as the cell membrane that we just look at is the first membrane, is the first layer, for some there's another layer beyond the cell membrane, which we call the cell wall. In plants, that layer is filled with a material called cellulose. In fungi, that material is filled with something we call chitin or chitin. In bacteria, it is filled with what we call peptidoglycan. As we go along, we'll get into the nitty-gritty of these. Now, for plants, where two cells of plants meet, there is a layer that tries to tell you that these are two different, they are not touching each other exactly. That layer is called the middle lamella or lamella. But although we've put some material in between to separate them, we keep a connecting, let me say something like a connecting tube between one cell and the other cell. That connecting tube is what we call the plasmodesmata. So let's go back to the drawing and look at the top here where I'm going to circle. You see something called plasmodesmata, like a passage, and you see a straight line that is called middle lamella. This is what I'm referring to. So the plasmodesmata allow things to be exchanged between this cell and the nearby cell just above it. What are the functions of the cell membrane? What are the functions of the cell wall? You say cell membrane's job is, as I said, to regulate the entry of substance in and out. And at the outer cover of most cells, it's holding or protecting what is inside, like the way your skin is protecting things inside you. It also delimits the content. That means things just can't flow out anyhow, like your tummy. Your intestine could have pushed out, or just, if somebody give you a cut, it just can't come out. But the skin, your skin at this moment is trying to prevent these things from coming out. It also serves as a barrier. Your skin, I'm just comparing to your skin. Your skin is serving as a barrier, separating the environment weather from your intestine. Same the cell membrane does. It separates the content of the cell from the environment. And it gives shape and flexibility to most cells. But what about the cell wall? Because an extra material laid with cellulose, chitin or chitin or peptidoglycan, they're a little bit rigid. And that rigidity gives some support or mechanical strength to the cell we are talking about. The same rigidity brings some protection. Because it's so hard for many certain things to enter easily. Let's say bacteria to affect it or something. Then it gives shape to most plant cells or those cells with the cell wall. Then one thing I'm supposed to know is that although it's an extra material outside, it does not react and does not close or open beyond what it does, like the way I explained the cell membrane. So for them, when something is passed, it does not decide, can you pass, you can't pass, unless maybe you are bigger than the size, but it does not interact with chemicals passing, substances pass, it doesn't react with you. So you just can pass through. It is the cell membrane that decides whether you can go through or not. So mostly we say that it is freely permeable to substance, so it does not impede things. Under plasmolysis and turgidity, when you study under osmosis, it is this same cell wall of plant that will resist the bursting of a cell, which brings about the turgidity in plants. So your screen is some differences between cell wall and cell membrane. Okay, let's move on to the next After cell membrane, the next thing usually you see is your cytoplasm. We say the cytoplasm is a jelly-like substance that fills the cell membrane and the nucleus. The nucleus, we are using the animal cell for this, is the central place thing. So the distance between the cell membrane and the nucleus, all that space is filled with a gel-like material, which we call cytoplasm. But the fluid itself is called the cytosol. Why did I use the word cytoplasm? The liquid is the cytosol, but within the liquid plays a lot of things, suspended with a lot of things. So the entire thing, or the mixture, and the places occupying all together is what referred to as the cytoplasm. And it's made up of 90% water. But this cytoplasm, which is a fluid, can flow. So when it's flowing, we call it cytoplasmic streaming. And once they flow like a river, if there are logs or certain things, a plank or a leaf on a river moving, at least to carry the leaf along. So cytoplasmic streaming also carries substances within the cell. Then we say cytoplasm also contains what we refer to as a living part and a non-living part, or structures. The living structures are referred to as the organelles. These organelles too, like we have a membrane outside, the same nature of the membrane tries to cover or enclose these organelles or structures so that one organelle can perform a certain function different from the other, or when they are performing their function, it does not spill over to the next place. So this is what the organelles do. So that means a cell itself, it's like a bag of chemicals that has been covered so that all reactions can take place in one and not move to the next. So the next can also take a place on its own without affecting the other. Now, what are the functions of cytoplasm? We said that all important organelles, every living thing and non-living thing are suspended in it. It stores vital chemicals and a lot of metabolic pathway or reactions take place. For the Form 3's listening, at least you've done glycolysis and Krebs cycle. Glycolysis takes place in the cytoplasm. Same when we start with photosynthesis. There's a portion of photosynthesis that takes place in the cytoplasm. And we say metabolism is what? The chemical reactions or pathways that take place in a cell. We divide it into anabolism and catabolism. While anabolism is building up and catabolism is breaking down. And all these things take place in the cytoplasm. Now, as I mentioned, there are some organelles. And we define an organelles as minute structures within an eukaryotic cell that are bounded by a membrane and perform a specific function. So because you are covered, we say you are a compartment. So we mostly say it ensures compartmentalization and division of labor. Why do we do that? Compartment means that chloroplast can do a job. At the same time, mitochondria too has its own compartment doing its thing. Lysosome has its own compartment doing something. In that case, it allows everybody to do part of the job that the cell needs. So we say there's division of labor. Now these organelles, as we just defined, listen to it, define as structures with membrane, but let's go down and explain it better. Once you have a function, a role they play and it's covered, we say it's a membrane. We have those that are double membrane, those that are single membrane. And we have those that don't have membrane too, but we still call them organelles. By the ribosome, the centrioles, and the cytoskeleton. Then we said that the living part of a cell is the protoplasm, is the nucleus and the cytoplasm together. Now let's zoom on one after the other on cells, the organelles. On your screen is a nucleus. You can see there's something called nuclear envelope. It has an outer side and an inner side. And note something called nuclear pore. It is the place that substances are exchanged between the nucleus and the cytoplasm. And a dark portion called nucleolus. And you can see some thread-like materials which we've labeled as DNA. But when they condense, thread-like actual material is called a chromatin. It's actually a DNA, but when they coil everything in the biology sense, we call it the chromatin. But when it condenses, and you can see it as two chromatids held at the central, so as they're held at the joint, then we are saying it's called a chromosome. And what is the function of a nucleus? Then there's a fluid, before I go on, there's a fluid in the nucleus we call the nucleoplasm. Now the function of the nucleus is to control the activities of the cell and also ensure protein synthesis. When we say it controls the activity of the cell, all that we are trying to say is that the DNA will send something in the form of mRNA, comes to the ribosome, they make proteins. The proteins may be used to make enzymes, hormones, whatever they want to send out. Or some are left within the cell to also undertake certain functions. Now, they are going to control the metabolic, enzymes are going to control the metabolic pathways in the cell. So indirectly, it is the nucleus who indirectly is controlling the metabolism, which controls the activities of the cell. So what is that? We say that it is the nucleus who indirectly is controlling everybody in the cell. And there's a proof that when you take a nucleus out of an amoeba for a week, most things stop, all activities stop. But when you put it back, the activities continue. It initiates cell division and also houses the genetic material, mostly that we pass on from parent to offspring. Now let's look at the mitochondria. Let's look at the drawing first. Mitochondria is the one on your left, on your screen. You can see that it has two parts, an outer membrane and an inner membrane. The outer membrane is smooth and the inner membrane is folded. The folded projection or cristae. Now there's a fluid inside the mitochondria. The fluid inside the mitochondrion is what we call the matrix. Then in the matrix are the mitochondrion having its own ribosomes, its own DNA, and some enzymes, so they can undertake its own activities. Sometimes it can divide even when the cell has not divided. So mitochondria can undergo its own cell division irrespective of the cell even dividing. And I compared it to the chloroplast on your right because something is so unique about these two. They have double membranes, and part of the membrane, once membrane fold in, once membrane forms stacks on each other. Then all of them have fluids, all of them have their own RNAs, all of them have their own DNAs, all of them have their own enzymes, and they undertake their own activity and can decide to divide irrespective when the cell has not even divided. So it's just because there's a theory of what we call endosymbiosis, how they got into a cell. They believe it were on their own, and they were brought into a cell by symbiosis, so they function as such. So back to notes, as I said, first you have to talk about the shape. It is double bounded, the outer is smooth, and the inner is projected into what we call the crista or cristae, and the fluid is called the matrix. Has a lot of enzymes that undergo what we call oxidative respiration that would help us produce energy. Energy is stored in the form of ATP. So mitochondria's function is to produce energy and also to store and later release them. Now, the counterpart, the chloroplast, is oval or disc shape. So in the exams, we look at how you describe the shape. And they're also double membrane. So nucleus is double membrane, chloroplast is double membrane, mitochondria is double membrane. And they have stacks. If you have coins, let's say you have about 10, 20 pesos, and you pack them in a stack form from down up, each of the coin, one coin will be called a thylakoid. One coin will be called a thylakoid. The 10 together as a stack, just a 10 pack. If you are trying to hold the 10 all in your hand, then that 10 you are holding is the granum. But if you have another set of 10 and you are referring to like 2 stacks or 2 piles, then in biology we say grana. Again, so if you have set of coins that if you arrange from up down in a vertical form, one of the coin is a thylakoid. If you have a pack of that 10 together and you are trying to refer to it in chloroplast study, we say that is the granum. If you have 2 of the 10s, 3 of the 10s, 4 of the 10s, then we say it's what we call the grana. And they are connected to each other by what we call the stroma lamella. The stroma is the interior or the liquid inside the inner membrane. The function is that they also utilize energy to produce food in a process called photosynthesis. Then we have vacuole and ribosome. Let's look at a picture of them. Okay, so the vacuole is something like a sac, something like a fluid, a sac that has fluid in it and is bounded by a single membrane. So that's a single membrane thing. Then there's a fluid in it. The fluid is called a cell sap. In plant study, mostly the membrane that, what we call the vacuolar membrane, in plants mostly we call the tonoplast, a name given to the tonoplast. But you've just been using it across board, but usually plant cells. Now this liquid is like a storehouse for storing excess fluid, excess ions, excess molecule, fat like food, waste, gases, and that's what it does. In plant cell, it plays a part in osmosis or turgidity formation or plasmolysis. Now on your right is the ribosome. These are tiny structures that the makeup is actually made from the DNA, pass into the nucleolus, they form over there and they are brought. What is passed into the nucleolus is the RNAs. Then the RNAs in the nucleolus would bond or wind around some proteins and they come out as ribosome, tiny structures. They are of 2 subunits, a big unit and a small unit. When they join, there's a gap in between them that where we put the mRNA or a certain molecule which help us to synthesize protein. So we say that platform they give, it is not them doing the synthesizing the protein, but it's just a place, a platform that allows the process to go on. So it is not ribosome producing protein, but it's the site or the platform for producing protein. So these are your notes. You can jot them down. Let's look at the ER that most students were struggling with. Let's look at a picture of this. So this is your nucleus on your far left. Just associated with it are these zigzag things around it with some red dots. And the same zigzag, some don't have any red dots on them. We call the red dots the ribosomes, which I just described, they are attached to some. So because of that, it appears as a rough structure. So we call the rough ER. And those with that is called the smooth ER. Now note what is there. I said that something is passed on from the nucleus, that's an mRNA, that comes to sit on the ribosome. Now the middle picture is showing you how a nuclear membrane pore passes things through it, and at the end it forms a vesicle, cuts out and fall onto another organelle called the Golgi body. Some say Golgi body or Golgi body. So what is the job of the endoplasmic reticulum? Its job is to allow transport of protein and other materials to other parts of the cell. It also connects the nucleus to the cell membrane. So the nucleus can send something it wants to go out, maybe straight out like that, without interfering with the, or getting interfered with the cytoplasm. Sometimes they will pass on whatever protein that they've made into the Golgi body we call the post office, because the post office, because plenty protein material are sent to it. It takes time, then sort them out, regroup them and classify them according to who become a hormone, an enzyme for this function, that function, and where you're supposed to go. So that is it. But you see that the Golgi body has some cap-like structure. It's like zigzag, but it's like a cap-like structure. We call it the cisternae. And on your far right, I've given a hand drawing that makes it easy. You can draw in exams. And the person who identified Golgi body is called Camillo Golgi. And as I said, they are involved with packaging, processing, and making membranes around things. So lysosome is formed, plenty of this protein has passed from the endoplasmic, because it was made by the ribosome, which is sitting in the endoplasmic reticulum, passed through it. Then it moves to the Golgi body. When it gets to the Golgi body, it selects those that are hydrolytic enzymes, pack them into one container, about 50 of them or more, and then they ship them out to go, and if it's, the lysosome will stay within the cell and work on the cell. But if it's a vesicle to be translated like a secretory vesicle, then they will have to push them out by exocytosis. And the scientist who identified Golgi body is called Camillo Golgi. It was named after him. So let's take something back on ER. We have said that the function of ER is to do. It's also a platform for ribosome attachment so that they can undertake protein synthesis. So the ribosome, we have two types. Those who are attached to the endoplasmic reticulum, they usually make proteins that will be transported out. If you are not attached, you make proteins that will be used within the cell. So we have free and attached ribosome. So attach, make things that may one day send it out. Then they collect and store synthesized material. They also make lipids. Now this lipid and steroid stuff are done by the smooth endoplasmic reticulum. Usually the rough don't do these ones, but the smooth does these things. They also make substance that will detoxify certain drugs or alcohol that we take into our cells. So I spoke about lysosome. They are small, they are spherical, they are single membrane. So Golgi single membrane, lysosome single membrane, vacuole single membrane. I remember I asked you a question at the top. Mention organelles that are double membrane, single membrane, and no membrane. So note them: nucleus double, mitochondria double, chloroplast double. ER double. You come to single, vacuole, lysosome, Golgi body are all single. Now these ones, as I said, was formed by the Golgi body, who has made a vesicle around digestive enzymes. And their job is to destroy worn out things within the same cell. Let's say mitochondria has finished its job and it's not functioning. Somebody has to clean the house. That's his job. So it destroys part of its cell content. Sometimes too, if the cell itself should die, they break open and destroy the cell that they were living in. And sometimes it's also believed that they can be sent out to the nearby cell and destroy a nearby cell if there's some malfunction with that cell. And content of those cells that are broken down are used by another cell. Then there's something we call phagocytosis. So the first one that they destroy part of the cell is called autophagy. If they destroy the entire cell they live in or another entire cell, we are talking about autolysis. Phagocytosis is when the cell itself has engulfed food, like we see in amoeba. But when the food enters the cell, the lysosome is going to go around that vesicle, engulf it again for the second time whilst it's inside the cell, and break it down. So why do they do that? When they clear off these debris in their body, it allows new ones to be made. If it's a whole cell that they destroy, it tells for a space to be created so that a new one can be replaced to that side. It also helps in repair of tissues and cells. The same lysosome, when you go to our main video, you see that when we were born, we had this thing like the toad's hind limb. But the lysosome tried to eat them out for us so that we have a free hand. Now the other structure too is called centriole. A centriole is a pair of microtubules, as you've seen on your screen. There are 9 of them. 3 each that goes round. So we say 9 triplets of rod-like structures. Now this centriole can be located very close to the nucleus as a clear zone. That clear zone is called the centrosome. But what is in the centrosome are these structures. And if one is standing up this way, the other one is perpendicular to it. So we say it's a pair. And prior to cell division, each pair would divide into 2. This one will reproduce another pair, and this one will produce another pair. So there will be 4. Then they would have to move to separate parts. And this is how they behave. So we say that when they go to the opposite poles of the cells, they will start to form spindle fibers, as we studied in cell division, hold on to the chromosome and try to pull them apart so that the telophase stage, they end and help in cell division. Now, one of the few organelles that of late we encourage to teach the cytoskeleton. It's difficult to see it with most microscopes because they are ground substance, they are the back. But they are like a mesh net that holds the whole cell together. So we say it's like a crisscross mesh net that passes through the entire cytoplasm. It is not bounded by a membrane because they are made of protein filament. You can make it when you need it and you can break it down when you don't need it. Usually we believe that even lysosome pass on these things to meet their target if a foreign substance enters the cell. We say it's the road network within the cell that things pass on to specific place they want to go. Now there are 3 of them. We call them the microtubules, the intermediate filament, and the microfilament. And as I said, their job is to give extra strength to the cell, maintain the shape of the cell, anchor its organelles, and drive the cell movement. In some organisms, there are cilia and flagella being produced. The same centrosome with the centrole that we saw. A similar type can be found close to the side or the membranes of certain cells, and from there, be able to produce microtubules in that direction, not into the nucleus, but to the outside. If they produce shorter forms, we call them the cilia. They produce longer forms, we call them the flagellum. And when we did the centrole, we said that 9 triplets, but here there are 9 of them, but they are pair. So 2 of that. So 2, 2, 2, 2, 9. Then in the center, we have another 2. So we say it's a 9 plus 2 arrangement. Microvilli are extensions which we'll look at. So for all that we've discussed, we have a summary on your screen. This is what you can summarize, study it well, and when they ask any question, you can answer. So a nucleus, the shape is spherical, the membrane is double, internal fluid is called nucleoplasm. Yes, there are some enzymes, and there are some unique structures there, and the major function. So with much knowledge of this, I'll let us use the last 10 minutes to try and answer our questions again from what we've seen so far. So you can call us on 0302 221 705 or 0302 221 706. So I'll bring the question of the day back and let us see with this information if we can answer. Okay, let's go to the animal cell first. You can call through 030 2221 705 or 030 221 706. With the information we have, try your best to see if you can identify most of the organelles we've studied so far. Our students, where are you? Try and call through. Let's see if we can use some of the organelles we've seen so far to answer the problem of the day, either question 1 or question 2. Elizabeth. Hello.
Hello.
Elizabeth. Yes, on your screen is an animal cell, and from what we studied so far, let's see if you can identify some of the organelles labeled I, IV, and stuff on your screen.
I is the smooth ER.
Okay. I is smooth ER.
I is the smooth ER.
Okay.
And I is mitochondrion.
Correct.
And I is the rough ER.
The rough, I is the rough ER, okay.
Yes. And IV is the smooth.
IV is the smooth, okay.
And then is
try your best, you're almost there. Okay.
Then is the Golgi body.
Wow, okay. You've done your best. Clap for yourself.
Thank you.
Okay. Let's make few corrections. Depending on what you see, the eye, some would hold on and say ends at the small dot there, which is the vesicle, but it's actually pointing to the Golgi body. The I, she got it right. It was mitochondria. The III, you can argue it is pointing to the nuclear pore, but look at the drawing well, because there are no dots on that particular drawing. We say it is the smooth ER. Then the I vein didn't point exactly to the darkened part. It points to the interior of the cell, so of the nucleus. So we say it's the nucleus. Then V, you can argue it points towards the ribosome, or it's pointing to a rough ER. So this is what we wanted for the day, and they asked you the functions for it. They name some enzymes that can be found in mitochondrion and some that can be found in the nucleus. So let's look at the point of the day 1. Okay, let's look at the last one. It says point to a centriole and point to a vacuole. So where will a centriole be? Anything very close to the nucleus. Then a vacuole be a plain thing that is from the big two things forming just under I. Now let's look at point of the day 1 that I gave and look at the answers. So I is your cell wall, II is your cell membrane, III is your cytoplasm, IV your chloroplast, and the chloroplast membrane is the tonoplast. Then VI is your vacuole, VII is your nucleus, and VII is your nucleolus. So your cell membrane, your cell wall, cell membrane, cytoplasm, your chloroplast, your nuclear membrane, vacuole membrane, you have your nucleolus and your nucleus. These are what they expected of you. All too soon, our Ghanaian students across the country, I won't say I'm so disappointed, but it's the first time we'll come back with cell 2, which will lead us into DNA and stuff. We also implore of you, if there are some topics you find it a problem, you don't understand, go to our Facebook or call in through the call segment, share your view of topics you want us to treat, and we'll take that as part of our revision of areas that you find problems with. Go to our comment and type on YouTube. Leave the topics that you have problem with. We will look at it and we'll prepare for our next meeting. If not, then the next topic that we'll be having is cell 2, that will cover tissues up to DNA, cell division, and protein synthesis. All too soon, our time is up. Thank you all for staying through these 2 hours. And do your best that you go to our section and just look at the question of the day. They'll keep playing it. And when we just end this show, it will be on your screen. Take time, go over your books. You see that you've been exposed. You think you are studying the bigger topics. Form 1 topic, it seems my students have forgotten a lot, especially when we started with the initial part. There were 6 slides, but we couldn't finish. So next time we go to the 3, then we would find answers. If you have problem, go to our comment, leave it, and we'll read it for you. Thank you very much. Till we meet again, I was your facilitator, Osei Kwame Amwaten, taking you through cell study. Bye-bye.